Hello everyone, I am glad to see you on the course Systematics of Lower and Higher Plants. And today we have the theme of video lecture, Systematics as a Biological Science. Also we will consider characteristics and classification of bacteria and algae, for example blue-greens, green algae, and hyphates. So goal number 1. We need to study of plant systematics meanings, terms and characteristics, features of plant systematics and characteristics, reproduction and systematics of the bacteria. The first question, what is botany? Botany is a scientific study of plants and plant-like organisms. It helps us understand why plants are so relatively important to the world. Plants start the majority of food and energy chains. They provide us with oxygen, food, and medicine. The taxonomy, systematics, and classification are terms with similar meanings. They are all about the overwhelming diversity of living organisms, for there are more than 2 million species. Phylogenetics is a more fashionable term. It emphasizes evolutionary history of taxonomic groups. This taxonomic organization is hierarchical. Most scientists accept 7 main levels of taxonomy, or ranks. The highest is kingdom, followed by phylum, class, order, family, genus, and lastly, species. In addition to 7 ranks mentioned above, plant taxonomy uses intermediate ranks like subfamily, subclasses, or suborder, when taxonomic structure is too complicated. Names of species are binominals, which consist of the name of genus and species epithet. Throughout the long history of taxonomy, too many names were given to the same taxon. Now we have almost 20 million names to describe 2 million species. These 80 million excess names are synonyms, which should not be used in science. To regulate the use of names, nomenclature codes were created. They code specificity, for example the rule of priority. When 2 names are given for the same group, only earlier name is valid. Consequently, it is recommended to list the author and the year of description along with the name. So, another important concept of nomenclature is the nomenclature type. Particularly, this means that every species name must be associated with a physical museum specimen. In botany, these museums are a collection of dried and pressed plants called herbaria. Type specimens are of immense importance because there are no labels in nature, and only these specimens will tell about real plants or animals associated with particular names. Systematic as a science have next points. The first, systematic is a descriptive science, traditional approach similar to astronomy and anatomy. The next science, systematics, is also an experimental science, nervous paradigm. Hypotheses about the relatedness of plants are developed and then tested by analyzing data. And last, systematics is the first science. Our receptors were great taxonomists. They had to be in order to survive. They collected foods, medicines, and etc. from the wild. In the not too distant past, botany was one of the major subjects in school. Sadly, the most civilized we were become the further from our roots that we have grown. And about plant classification is based on botanical type and values or geographical or ecological communities. The next information about bacteria. Bacteria are microscopic, single-celled organisms that thrive in diverse environments. These organisms can live in soil, the ocean, and inside in the human gut. Bacteria share a number of common structures that are briefly described. Slime or extracellular polysaccharide. This is extracellular material loosely associated with the bacteria that is elaborated by some bacterial species that facilitated colonization of smooth, prosthetic surface such as intravascular catheters. The next, capsule. This polysaccharide outer coating of the bacterial surface often plays a role in preventing phagocytosis of bacteria. And last, peptidoglycan or cell wall provides bacterial shape and rigidity. The cell wall consists of alternating units of acetylglucosamine and acetylmuracil acid with N. The polysaccharide chains are cross-linked by a peptide bridge. It is a primary target of antimicrobial therapy because it is specific to prokaryotes. About reproduction, the most bacteria multiply by a process called binary fission. In this process, a single bacterial cell, called the parent, makes a copy of its DNA and grows larger by doubling its cellular content. The cell then splits apart, pushing the duplicated material out and creating two identical daughter cells. Some bacterial species, such as cyanobacteria and Formicotes, reproduce via budding. In this case, the daughter cells grows as an offshoot of the parent. Each starts at a small knob, grows until it's the same size as its parents, and split off. In this slide, you can see classification of bacteria, which include cocci, bacilli, and others with different shapes and forms. The next goal, we need study of characteristic, reproduction, and systematics of the blue-greens. Fossil history of the blue-greens extends back to the Precambrian. They are found in water, on soil, on rocks, and in the atmosphere. Habitat temperatures ranges from 0 to 85. Some species occur in lynx, liverworts, water ferns, cycads, and flowering plants as symbiotic. The microscopic blue-greens have no membrane-bound nucleus. The cell has muciglatulous sieve of peptine that surrounds the cell wall and inner cellulose wall. Cell contents, or for example protoplasm, include nuclear material with DNA fibrils, chlorophyll A, and accessory pigments of blue, red, orange, and xanthophylls or yellow color. These pigments are involved in capture of light and subsequent formation of carbohydrates. Cyanophyllene starch is a storage product. Blue-greens forms may be one cell or colonial or filamentous forms. They have no thread-like structures, flagella for movement. About reproduction. Blue-greens have no known sexual reproduction. The reproduction of unicellular forms is by cell division, in which the cell wall folds and enriches the cell into cells. Colonial and filamentous forms fragment into separate species. Filamentous forms have various specialized cells. An akinete is a resistant cell filled with food reserves, which can determinate to form a new filament, and transparent heterocyst functions in nitrogen fixation and frequently is located where the filament breaks. Some genera have exospores. These are cells that branch off the filament. On this slide, you see classification and species from blue-greens. For example, Glogeocapsa, Oscillatoria, Anabaena, and Nostoc. The next goal, number 3, the study of characteristics and features of algae and the study of characteristics, reproduction, and systematics of the green algae. General about algae. There you can see morphological types of thallus differentiation, which include monadic, amoeboid, palmuloid, coccoid, filamentous, multifilament, lamellar, siphon, and siphon-clad types of thallus differentiation. And also you can see reproduction of algae with vegetative, asexual, and sexual forms. Reproduction processes, when we talk about vegetative reproduction, in which parts of the thallus are separated without any noticeable changes in the photoplast. For example, fragmentations. The next, asexual reproduction. It is carried out through special spores. for example, zoospores and aplanospores. And last, sexual. This process in algae can occur with participation, for example, isogamy, heterogamy, oogamy, or without the participation of gametes, for example, hologamy and conjunction. Green algae constitute a very ancient group of plants with fossil occurring back to early period of the Paleozoic era. They are mainly freshwater plants, but also occur in salt water, in soil, on tree bark, and on snow. Green algae forms are unicellular, colonial, filamentous, membranous, and tubular. The cells have many of the organelles found in cells of higher plants, including a vegetal wall, a nucleus with one or more nucleoli, a larger vacuole well-organized chloroplast, pigments of chlorophyll A and B, carotenes and xanthophylls, self-feeding nutrition or autotrophic form, and pure noids associated with starch formation. Motile forms usually have two whiplash flagella of equal length, and there are many various types of asexual and sexual reproduction. On this slide you can see classification of green algae, which include the next species: Volvox, Ulotrix, Cladophora, and Spirogyra. The last goal, number 4, the study of characteristics, reproduction, and systematics of the harophytes. The harophytes division has about 300 species, from mostly in freshwater with clean and calm water, usually forming extensive fricates, with a continuous carpet covering the bottom. For many of them it is necessary high content of lime in the water. Some harophytes can live in brackish water. Harophyta is the largest of freshwater algae. The length of thallus is usually 20 or 30, but can reach 1 or 2 meters. Thallus of harophytes have kind of brushy brushing filamentous or stem-shaped green-joined whorled shoots growing at the bottom of reservoirs, with multiply thin colorless rhizoids. On the main shoots are whorls of short lateral shoots, also joint structure. Growth of stems is apical, unlimited. The leaves have marginal growths. The leaves can be axial and fork separated. A set of pigments is similar to these in green algae, with chlorophyll A and B, the carotenoids, and more, and as production of assimilation also produced starch. Harophyta is characterized by sexual and sexual reproduction. Vegetative preparation occurs by rooting and subsequent growth of parts of the main shoots, or with the help of special unicellular or multicellular modules. The same purpose is served by special processes or secondary shoot growing from the old leaf, nodes, and nodulus. Sexual reproduction occurs with the help of the genitals, anteridium and ovum. On this slide you can see the main structure of Haraflagellus. So, we have considered full material in the framework of our video lecture. Thanks for your attention and goodbye.